It's pretty wild to think about, but you are legally blind for over an hour every single day.
Yeah, it really is. And you don't even notice it, right? Your brain completely hides it from you.
Exactly. I mean, right now, if you are listening to this while driving down a highway, your visual system is actively shutting off its data feed multiple times a second.
Right. We process the visual world as this continuous, stable, high definition stream.
But that continuity is basically a profound biological illusion. It's masking this sensory and motor landscape that is built on, well, these violent ballistic jumps and massive real-time computational suppression.
It is, I'd argue it's the ultimate biological magic trick.
I totally agree.
Because what we experience as a fluid, cinematic rendering of reality is actually being counted by this microscopic sensor just darting erratically across our environment.
Right, relying on a Brainstem engine that's running highly advanced calculus just to keep the image from blurring into a nauseating smear. So, welcome back to our ongoing deep dive into the neuroscience of movement. And for those of you tracking along with our overarching progression through the motor system, specifically, reflecting the foundational frameworks of Part 5 in Kendall's Principles of Neural Science, we are continuing our exploration of motor control today.
But we're pivoting, right?
Yeah, we're pivoting to perhaps the most mathematically precise motor system in the mammalian body, which is gaze control.
Which is just a fascinating system. And, given the focus on high-level neurobiology and knowing that many of you are researchers or PhD candidates.
Right. We get a lot of messages from you guys.
Yeah. So there is a really strong chance that you are listening to this behind the wheel of a car, navigating this complex, high-speed, three-dimensional environment right now.
Which is basically a setting that requires continuous, flawless spatial updating. So because of that, we're going to pace this discussion deliberately.
Yeah, we're taking our time with this one.
We want this to be unhurried. We are dealing with highly complex anatomical circuits and electrophysiological concepts, and we really want to let the sheer scale of this neural architecture sink in.
So to truly grasp the neurobiology of gaze, we first have to confront the fundamental, punishing biological constraint that forced this whole system to evolve in the first place.
Right, and you're talking about the fovea.
Exactly, the central pit of the macula.
Right, so we are dealing with an area of the retina that is less than 1 millimeter in diameter.
Which is incredibly tiny.
It is. It's the only patch of photoreceptors that is dense enough and with the necessary one-to-one ganglion cell convergence to actually provide sharp, high-acuity vision.
Right, and because of that microscopic size, the fovea covers less than 1% of our visual field.
Which means to read a dashboard dial or, to track a merging car, we are forced to move this tiny sensor with extreme velocity and pinpoint accuracy.
We are effectively viewing the universe through a moving keyhole.
That is a perfect way to put it, a moving keyhole.
And, well, to move that keyhole, we have to rely on the mechanical actuators in the orbit.
The hardware, basically.
Right, the hardware. And it's elegant precisely because of its relative simplicity compared to, spinal motor system.
Oh, for sure. We have no joints to stabilize here.
Exactly. No joints and no gravity to fight in the same way a limb does, right?
Right.
We're dealing with just 12 extraocular muscles total, so that's 6 per eye, and they are arranged in 3 antagonistic pairs.
Let's break those down slowly.
Sure. So first you have the horizontal pair, which is the medial and lateral rectus.
Okay, medial and lateral rectus, got it.
Then the vertical pair, the superior and inferior rectus.
Right.
And finally the torsional pair, which is the superior and inferior oblique.
Okay, so the anatomical origin of these muscles really dictates the complexity of their control vectors, doesn't it?
It completely dictates it.
Because the 4 rectus muscles, along with the superior oblique, they all anchor at the annulus of Zen. Right.
Yes, that fibrous ring at the orbital apex.
Right. And they travel anteriorly to insert onto the sclera.
Mm-hmm.
But the superior oblique kind of breaks from the straightforward geometry, which creates this distinct biomechanical challenge.
It really does. It's such a weird anatomical setup.
Explain how that works, because it's basically a pulley, right?
Yeah, exactly. So the tendon of the superior oblique travels anteriorly, but before it reaches the globe, it threads through the trochlea.
And the trochlea is just this cartilaginous ring at the supromedial aspect of the orbit.
Right. It acts literally as a biological pulley. So the tendon passes through it, makes an acute U-turn, and reflects backward and outward to insert onto the superior posterior quadrant of the globe.
Wow, okay, so it goes forward, hooks through the loop and pulls back.
Exactly. And because of this pulley, the muscle's effective origin, like the functional point of traction, is the trochlea itself.
Not the annulus of Zinn where it actually anchors.
Right, not the annulus.
That's wild. So this pulley system means we are dealing with a highly nonlinear biomechanical plant.
Highly nonlinear. The action of any single muscle is not fixed at all.
Meaning its force vector is entirely dependent on the eye's starting position in the orbit.
Exactly. Let's take the superior rectus as an example.
Okay, let's do that.
So its line of action is offset from the visual axis by about 23 degrees when the eye is in primary position, like looking straight ahead.
Okay.
If the eye is abducted by 23 degrees, so looking out toward the ear, aligning the visual axis with the muscle plane, the superior rectus acts as a pure elevator. It just pulls the eye straight up.
But if the eye is adducted, right, like rotated medially toward the nose.
Right. If you look inward, that vector geometry completely shifts.
Because the angles have all changed.
Exactly. The superior rectus now pulls almost orthogonal to the visual axis.
Oh, wow.
Yeah, so in that adducted state, contracting the superior rectus primarily causes intorsion, rotating the superior pole of the cornea toward the midline.
So it's not even elevating anymore.
Barely. Elevation in that adducted state has to be taken over mostly by the inferior oblique.
Okay, wait, so if I tilt my head, it's kind of like, think of a camera mounted on a really complex 3D gimbal, right?
That's a great analogy, actually.
Like if I tilt the gimbal, the camera has to rotate on its own axis just to keep the horizon level. Is that essentially what torsional movement is?
That's exactly what it is. Intorsion and extorsion.
Yeah.
And the nervous system has to constantly solve these shifting vector matrices in real time.
Just to keep the horizon stable.
Right, and it's governed by principles like Listing's law to ensure we don't introduce unwanted torsion during oblique gaze shifts.
And it has to solve this matrix for two eyes simultaneously, which is just staggering.
Yeah, it has to maintain stereopsis. We require perfect binocular yoking.
Right, Herring's law of equal innervation.
Exactly. Herring's law mandates that conjugate eye movements are driven by simultaneous equal innervation to yoke muscle pairs.
So meaning a saccade to the right demands equal drive to the right lateral rectus and the left medial rectus.
Right. They have to move perfectly in sync or you see double.
Okay, so the physical execution of this yoking happens at the level of the brainstem nuclei. But before we ascend into that circuitry, I think we should examine the consequences of losing one of the output pathways.
I agree. It's the best way to really understand the hardware.
Right. The segregation of these muscles across three cranial nerves, the abducens, trochlear, and oculomotor, it really provides a remarkably precise clinical map of this motor plan.
It does. It gives us a window into how the software talks to the hardware.
So let's trace the clinical presentation of an abducens, or cranial nerve 6 lesion.
Okay, so cranial nerve 6. The abducens nucleus sits in the caudal pons, and it innervates only one muscle, the lateral rectus.
So just the muscle that pulls the eye outward.
Exactly. So a peripheral lesion here paralyzes that muscle, which severely limits the abduction of the ipsilateral eye.
The eye on the same side as the lesion.
Right. And the immediate perceptual result for the patient is uncrossed horizontal diplopia double vision.
And that gets worse when they try to look toward the side of the lesion, right?
Yes. Because the intact contralateral medial rectus continues to pull the healthy eye inward.
But the paralyzed lateral rectus can't pull the affected eye outward to match it.
Precisely. The visual axes diverge, and the foveas receive disparate images. You see two of everything.
Now, the oculomotor nerve, cranial nerve the third, that represents a much more catastrophic failure mode, doesn't it?
Oh, absolutely. It's the heavy lifter. It emerges from the midbrain and carries motor fibers for 4 extraocular muscles.
The medial rectus, superior rectus, inferior rectus, and inferior oblique.
Right. Alongside the levator palpebrae superioris, which elevates the eyelid.
And it doesn't just do somatic motor, right?
No, it also carries the preganglionic parasympathetic fibers to the ciliary ganglion, which governs pupillary constriction.
Okay, so when a complete cranial nerve 30 palsy occurs, which is often due to compression from a posterior communicating artery aneurysm.
Yeah, that's a classic cause. The clinical triad you see is unmistakable.
Walk us through it.
You see severe pactosis, which is a drooping eyelid due to levator paralysis. You see a fixed, dilated pupil from the loss of parasympathetic tone.
And then there's that classic down and out resting position of the globe.
Right. The eye literally just sits aiming down and outward.
But wait, let's unpack that, because that resting position perfectly illustrates the concept of continuous dynamic tension in the orbit.
It really does.
Because if the oculomotor nerve is severed, why does the eye actively turn down and out? Like, if the power's cut to most of the muscles, shouldn't it just rest in the middle?
It's a great question, and it's a huge misconception. The eye doesn't just float to a neutral center when cranial nerve 3 is cut.
Because it's a tug of war.
Exactly. The denervation eliminates the baseline tonic firing to those 4 muscles, effectively removing them from the orbital tug of war.
So the ones that are left over just take over.
Right. The moment that opposing tension is removed, the baseline tonic firing of the surviving nerves just dominates.
So the lateral rectus, which is cranial nerve 6, is still fine.
Yeah, and it physically drags the eye outward into abduction.
And the superior oblique from cranial nerve 4 is also fine.
Right, and that drags the eye downward and intorts it. So that down and out position is not a passive droop.
It is an active deviation driven by unopposed, healthy muscles.
Exactly. Which brings us to the trochlear nerve, cranial nerve 4.
Which innervates only the superior oblique.
Yes. And this is an anatomical oddity. It is the only cranial nerve that exits the dorsal aspect of the brainstem and decussates crosses over entirely before innervating its target.
So a lesion here removes the primary intorter and depressor of the adducted eye. What does the patient experience?
While the unopposed inferior oblique causes the affected eye to drift upward and extort, rotating the visual field outward.
Which has to be incredibly nauseating.
Oh, it causes a vertical and torsional diplopia that is profoundly disorienting.
But there is a behavioral compensation for this, right? It's fascinating.
It is. The central nervous system will execute this purely mechanical workaround. Patients will present with a chronic compensatory head tilt.
Away from the side of the lesion, correct.
Exactly. And this is the basis of the Gielschowski head tilt test in the clinic.
Okay, explain how the head tilt actually fixes an eye muscle paralysis, because it sounds crazy.
It does, but it's brilliant. By tilting the head toward the healthy shoulder, the vestibular system detects the roll and triggers the vestibulo-ocular reflex to counter-roll the eyes, trying to keep the horizon level.
Right.
This head tilt forces the healthy eye to intort, which signals the damaged eye that it needs to extort.
Oh, I see.
By structurally keeping the head in a position that demands extorsion from the damaged eye, the brain minimizes the need for the paralyzed superior oblique, the intorter, to fire at all.
So the patient literally manipulates the geometry of their cervical spine to bypass a broken muscle in their eye.
Yeah. It demonstrates the absolute primacy the brain places on maintaining foveal alignment. It will twist your neck all day just to keep your vision single.
That is just incredible. Okay, so with the mechanical plant and its output pathways mapped, we can look at the actual software.
Right, the functional classes of eye movements generated by the brainstem and cortex.
Because while they all converge on those same motor nuclei we just discussed, their sensory triggers and neural pathways are distinct.
Very distinct. We can broadly divide them into gaze-shifting mechanisms like saccades and vergence, and gaze-stabilizing mechanisms.
Like the vestibulo-ocular reflex, optokinetic nystagmus, and smooth pursuit.
Right. And we must also include fixation in that stabilizing category.
Let's talk about fixation, because I feel like it is heavily misunderstood.
It really is. People think of fixation as a passive resting state, just a simple absence of movement commands.
Like the engine is idling.
Right. But in reality, maintaining gaze on a stationary target is an intensely active, metabolically demanding process.
Because the oculomotor system is inherently noisy.
Extremely noisy. Without active suppression, the eyes would constantly drift off target due to fluctuations in muscle tone and neural firing.
So how does the brain actually clamp that down, electrophysiologically speaking?
It relies heavily on the omnipause neurons.
Right, the omnipause neurons. Those are located in the nucleus raphe interpositus, in the midline of the pons.
Exactly. And these neurons exhibit constant tonic firing during fixation. They release potent inhibitory neurotransmitters, primarily glycine and GABA.
Directly onto the burst neurons that generate saccades, right?
Yes. They clamp them in a hyperpolarized state.
Okay, so to see the necessity of this inhibitory clamp, we just have to look at opsoclonus.
Right, opsoclonus. It's a rare condition, often secondary to paraneoplastic syndromes or viral encephalitis.
What happens when a patient gets that?
When the omnipause neurons or their cortical inputs are damaged, that inhibitory clamp is lost.
The brakes are cut.
Exactly.
Yeah.
The burst neurons just fire spontaneously and chaotically. The patient suffers from continuous, involuntary, multidirectional saccades.
Which means their visual acuity just plummets.
Yeah, because the fovea is never allowed to dwell on a target long enough for the visual cortex to process the image. The world is just a vibrating blur.
That sounds terrifying. But it highlights that active fixation is the absolute prerequisite for our most heavily utilized gaze-shifting mechanism, which is the saccade.
Right. Saccades are the rapid ballistic jumps we use to actively sample the high salience features of our environment.
And the kinetics of these movements are just staggering. I mean, a large saccade can reach peak angular velocities approaching 900 degrees per second.
And the entire movement is completed in 30 to 50 milliseconds. It's violent.
Alfred Yarbus did some foundational eye-tracking experiments on this back in the 1950s and 60s, didn't he?
He did. His work definitively showed how we deploy these jumps. We don't sweep a scene raster style, like a printer or a scanner.
Right.
When looking at a human face, for instance, our saccades jump relentlessly between the eyes and the mouth.
Which makes sense, because those are the nodes of highest social and emotional information.
Exactly. The intervening space, like the cheeks, is essentially ignored. We just bounce from eye to mouth.
Now, because these movements are completed in roughly 40 milliseconds, there's zero possibility of real-time visual feedback, right?
Zero. Visual processing from the retina to the primary visual cortex and back down to the motor centers takes upward of 70 to 100 milliseconds.
So the eye movement is literally over before the brain even registers that it has begun.
Exactly. And that defines the saccade as a purely open-loop ballistic command.
Meaning the brain must perfectly pre-calculate the amplitude and direction of the required vector before releasing the inhibitory clamp on the omnipause neurons.
It has to have all the math done before it fires. And crucially, during that 40-millisecond flight, the visual system undergoes saccadic suppression.
This is that period of legal blindness we mentioned at the very start of the episode.
Yes.
Because if the retina were actively transmitting during a 900 degree per second sweep, the resulting motion blur would be catastrophic to our spatial perception.
It would be like whipping a video camera back and forth. You'd just see streaks.
So how does the brain shut it down?
The brain utilizes a corollary discharge.
Okay, explain that.
It's essentially a copy of the motor command. It's sent from the superior colliculus up to the visual cortex, likely via the medial dorsal thalamus.
And what does that copy do?
It actively suppresses visual sensitivity, particularly in the magnocellular pathways, which are responsible for motion detection.
So we are functionally blind during every single saccade.
Completely. The stability of our visual world relies entirely on the brain's ability to stitch together the higher resolution snapshots taken during the fixation pauses, and it just completely discards the noisy data from the transit phase.
That is just, it's unbelievable computing power.
It really is.
So let's descend into the brainstem to examine how that transit phase is mathematically generated.
Down to the engine room.
Right. Because the pre-calculation of a saccade requires overcoming the specific biomechanical properties of the orbital plant.
And the physics here are challenging.
Because the globe is suspended in orbital fat and connective tissue, right? Which presents a massive viscous drag.
It's like trying to move something through thick syrup. Moving it rapidly requires a massive spike of force.
But once it reaches its new eccentric position, the elastic restoring forces of the stretched antagonist muscles and fascia want to pull it back to center. Like a rubber band.
Exactly. You have to fight viscosity to get there and elasticity to stay there.
So this necessitates the famous pulse step sequence of innervation.
Yes, the pulse step. If we record extracellularly from an abducens motor neuron during a horizontal saccade, we see a really dramatic firing pattern.
Walk us through the timing of that.
So about 8 to 10 milliseconds before the eye actually begins to move, the neuron erupts into a high-frequency burst. This is the pulse.
How high of a frequency are we talking?
We are talking firing rates scaling linearly with saccadic velocity, easily hitting 600 to 1,000 action potentials per second.
Wow. And this explosive burst provides the force necessary to overcome that viscous drag in the orbit.
Exactly. It shoves the eye through the syrup.
But the pulse alone would result in the eye sliding right back to center the moment the burst ends.
Right.
Because of the elastic pull.
Right. So to hold the new position against those elastic restoring forces, the motor neuron must transition immediately from that extreme high frequency burst down to a new, steady, tonic firing rate.
And this rate has to be higher than its initial baseline.
Yes. And the sustained elevated rate is the step.
It reminds me of, imagine you are underwater trying to push open a heavy vault door that has a massive spring attached to it.
Okay, I like that.
The pulse is the explosive full-body shove you have to give it just to get the door moving through the water. That's the viscosity.
Right.
And the step is the constant heavy leaning you have to do to keep the door open against the spring. That's the elasticity.
That is a perfect analogy. And the ratio of that pulse to the step determines the kinematics of the saccade.
But what is truly remarkable here is that the higher cortical and midbrain centers initiating the movement, like the frontal eye fields or superior colliculus, they don't calculate both parts.
No, they only encode the velocity and amplitude command. They only transmit the pulse.
So the brainstem itself must calculate this step.
Yes, the brainstem has to do the calculus.
The biological neural integrator.
Exactly. The network takes the velocity command, the pulse, and mathematically integrates it with respect to time to generate the position command, the step.
That is just phenomenal. Where physically does this integration happen?
Well, the physical location depends on the axis of movement.
Okay, let's start with horizontal.
For horizontal saccades, the burst neurons are located in the paramedian pontine reticular formation, or PPRF.
PPRF, right.
They send their velocity pulse directly to the ipsilateral abducens nucleus. But they also send collaterals to a network of cells primarily located in the nucleus prepositus hypoglossi and the medial vestibular nucleus.
So this prepositus vestibular network acts as the horizontal integrator.
Yes. It receives the transient high-frequency burst and, through highly complex network dynamics, which likely involves, reverberating recurrent collateral circuits, and the slow decay kinetics of NMDA glutamate receptors.
It's basically a feedback loop holding on to the signal.
Exactly. It transforms that transient burst into a sustained tonic output. It then feeds this newly calculated positional step back to the abducens motor neurons.
And vertical saccades, they utilize an entirely separate zip code in the midbrain, don't they?
They do, but it's an analogous setup. The burst neurons for vertical and torsional movements reside in the rostral interstitial nucleus of the medial longitudinal fasciculus, the RIM-MLF.
The RIM-MLF. Got it.
And their velocity signals are integrated by the interstitial nucleus of Cortical, which provides the step command to the oculomotor and trochlear nuclei.
The perfection of this biological calculus is astonishing. But it's not infallible, is it?
No, it's not. The neural integrator is inherently leaky. Yeah. Even in a completely healthy system, the reverberating circuits cannot hold a perfectly steady firing rate indefinitely without visual feedback.
So if you place a healthy subject in complete darkness and ask them to hold an eccentric gaze-like, looking hard to the left, what happens?
Their eyes will slowly drift back toward the primary position. The math decays.
And when the integrating networks are damaged, say due to cerebellar degeneration or specific brainstem lesions or even just intoxication from sedatives or alcohol, that integrator becomes excessively leaky.
Right. The step command decays rapidly.
So what does that look like clinically?
The patient attempts to look to the side, the pulse gets them there perfectly fine, but the failing step cannot hold the eye against the elastic tissue forces.
So the eye just slides back.
The eye drifts smoothly back toward the center, which is then followed by a quick corrective saccade back to the target.
And then it slides again.
Exactly. This alternating slow drift and rapid correction is the hallmark of gaze-evoked nystagmus.
Okay, so the brainstem engine, the burst neurons in the PPRF and RIM-MLF, the integrators, the on-to-pause clamp, that all provides the raw mechanics of movement.
Right, the engine block.
But the PPRF and RIM-LOF are essentially blind. They only execute the math. They rely entirely on higher-order structures to define the actual target coordinates.
Yes, someone has to tell them where to look.
And the primary interface, bridging that cognitive intention and brainstem execution, is the superior colliculus in the dorsal midbrain.
The superior colliculus is a masterpiece of topographic alignment. It's organized into these very distinct layers.
Let's unpack the layers because they map perfectly onto each other, right?
Yeah. The superficial layers are exclusively sensory. They receive direct input from the retina and primary visual cortex.
And they maintain a strict retinotopic map of the contralateral visual field.
Right. So a stimulus in your upper left visual field activates a discrete patch of neurons in the medial anterior portion of your right superior colliculus.
Okay. And then beneath these visual layers lie the intermediate and deep motor layers.
Yes. These layers contain the output neurons that project directly down to the brainstem burst generators.
And crucially, the motor layers contain a movement map that perfectly registers with the visual map right above it.
It's a one-to-one mapping. If you lower a stimulating electrode into the deep layers and apply a microcurrent, you don't elicit just some random eye twitch.
You get a specific vector.
You elicit a saccade of a highly specific amplitude and direction. The colliculus operates on population coding.
Meaning a target doesn't just activate a single motor neuron.
Right. It activates a broad hill of neuronal activity. And the spatial location of the peak of this hill on the collicular map dictates the precise vector of the resulting saccade.
So the superficial visual layer detects the target, passes the signal straight down in a columnar fashion to the deep motor layer, and the motor hill triggers the PPRF.
It is a hyper-efficient sensor and motor reflex arc.
But, and this is a big but, an organism operating solely on a collicular reflex would be a complete slave to its environment, wouldn't it?
Oh, totally. You'd be constantly orienting toward every flashing light, every sudden movement in your periphery.
You'd be utterly incapable of sustained task focus.
Right, so the system demands a powerful inhibitory override.
And that override is provided by the basal ganglia, specifically the substantia nigra pars reticulata, or SAR.
Yes. The SAR serves as this potent gatekeeper. Its neurons are tonically active, they fire continuously at high rates, and project massive GABAergic inhibitory synapses directly onto the deep motor layers of the superior colliculus.
So this tonic inhibition keeps the collicular motor map firmly suppressed. It's putting a leash on the reflex.
Exactly. It prevents the reflexive execution of saccades to random environmental noise.
So to break that suppression and initiate a voluntary goal-directed saccade, we have to look up at the cortical command centers.
We have to remove the leash.
Right. Let's trace the signal originating in the frontal eye fields, or FEF, which is located in the prefrontal cortex.
The FEF plays a dominant role in complex, memory-guided, and volitional saccades.
So when the FEF determines a target is actually salient and we want to look at it, what does it do?
It projects down to the caudate nucleus in the striatum. The caudate is essentially the input station of the basal ganglia.
And the caudate neurons are transiently activated, sending GABAergic projections to the SNR.
Right, so this creates a classic basal ganglia direct pathway. You are inhibiting an inhibitor.
So by silencing a specific population of SNR neurons, the caudate removes the GABAergic tone on a very specific patch of the superior colliculus.
This disinhibition opens the gate. The collicular motor hill can now fully form and fire its burks down to the brainstem.
So the FEF effectively sculpts the collicular output by selectively releasing the basal ganglia's leash.
Exactly. It points the flashlight, essentially.
We see the necessity of this prefrontal control really clearly in the anti-saccade task in a clinical setting.
The anti-saccade task is a brilliant neurological test.
Because it forces a battle between reflex and control. You instruct a patient to fixate on a central point. A bright target flashes to their right, but the instruction is to immediately look to the left, the exact opposite direction.
It is a literal battle between the reflexive collicular drive and cortical suppression.
Because the sensory flash triggers the superficial layers of the colliculus, urging a reflex saccade to the right.
And the prefrontal cortex must rapidly enhance the SNR inhibition to suppress that reflex, while simultaneously computing the inverted vector and disinhibiting the leftward motor map.
That is so much processing in just a few milliseconds.
It is. And patients with frontal lobe lesions or conditions like schizophrenia that affect prefrontal circuitry, they struggle immensely with this.
They just can't stop themselves from looking at the flash.
Right. They demonstrate high error rates, reflexively glancing at the target before correcting themselves, which exposes the loss of top-down executive control.
Okay, so while the FEF is deeply involved in the motor preparation and execution, the determination of what is actually worth looking at in the first place involves the parietal lobe.
Specifically, the lateral interparietal area, or LIP.
Right, because the parietal cortex is the master integrator of spatial information.
It is. The LIP constructs a dynamic, continuously updating priority map of the environment.
And how is that different from the visual cortex?
Well, the visual cortex encodes the physical properties of a stimulus like contrast or orientation or color. The LIP encodes salience and intention.
So it cares about whether the object actually matters to you.
Exactly. Single-unit recordings in awake, behaving primates demonstrate this beautifully.
What do those recordings show?
If a distractor flashes in the periphery, an LIP neuron might fire a brief, low-frequency burst just registering its presence.
Okay.
But if the monkey has been trained to specifically attend to that spatial location, or is holding that location in working memory to guide an upcoming saccade in total darkness.
That same LIP neuron reacts differently.
Completely differently. It will fire vigorously and continuously. The firing rate represents the behavioral relevance of the spatial coordinate, not just its sensory presence.
It's mapping importance, not just light.
Exactly. And when the parietal lobe is damaged, particularly in the right hemisphere, that priority map just collapses.
Which manifests as contralateral hemispatial neglect.
Yes. The patient isn't blind in the left visual field. The primary visual cortex is perfectly intact.
And the motor pathways to look left, the FEF, the colliculus, the PPRF, they are completely functional too.
Right. But the parietal map encoding the concept that the left side of space contains anything worthy of attention has been erased.
So the patient will ignore food on the left side of their plate or fail to shave the left side of their face.
The spatial landscape simply ceases to exist in their behavioral repertoire. It's profoundly strange.
It really is.
Yeah.
Now, up to this point, we have constructed an immensely powerful system designed solely to rapidly shift the fovea to a static spatial coordinate.
Right, a stationary point.
But the real world is kinetic. If you're driving and a bird flies across your field of view or a car merges into your lane, a ballistic open-loop saccade is insufficient.
Yeah, because if you only had saccades, you would have to constantly execute a series of jagged, corrective jumps to track it.
It would look like a strobe light. So, to stabilize a moving image on the fovea, we rely on smooth pursuit.
Smooth pursuit is fundamentally different from a saccade. A saccade computes target position. Smooth pursuit computes target velocity.
So it's a closed-loop system.
Exactly. It continuously monitors the motion of the image across the retina, which is called retinal slip, and generates a motor command to perfectly match the rotational velocity of the eye to the angular velocity of the target.
And the sensory arm of this reflex heavily depends on the middle temporal visual area, MT, and the medial superior temporal area, MST.
Right, because these cortical regions are specialized for processing complex visual motion.
They extract the direction and speed of the target and then project this velocity vector down to the brainstem.
Specifically via the pontine nuclei.
But matching eye velocity to target velocity perfectly in real time, that requires immense continuous calibration.
It does, and this is where the cerebellum becomes absolutely critical, particularly the flocculus and the dorsal vermis.
Because the cerebellar circuitry receives that cortical velocity command, but it also receives a massive influx of proprioceptive and extraretinal feedback, right?
Yes. It gets data regarding the actual velocity of the eye in the orbit.
So it's constantly comparing what you want to do with what you're actually doing.
It's an error correction mechanism. If the eye is lagging behind the target, so experiencing retinal slip, the Purkinje cells in the flocculus dynamically modulate their inhibitory output to the vestibular nuclei.
Fine-tuning the drive to the extraocular motor neurons to accelerate the eye and minimize the error.
Right. Without the cerebellar calibration, smooth pursuit breaks down completely. It revites into a choppy, stair-step pattern of corrective catch-up saccades.
So, smooth pursuit tracks motion across the x and y axis. But we live in three-dimensional space.
We do.
When an object approaches us along the z-axis, moving closer to the face, the requirements shift dramatically because saccades in pursuit are conjugate movements. Both eyes rotate in the same direction.
Right. But tracking an approaching object requires a disjunctive movement.
Vergence.
Vergence. As the object gets closer, the geometric angle between the two visual axes must increase to keep the image perfectly centered on both foveas.
The medial recti of both eyes must contract simultaneously, causing the eyes to converge inward.
And vergence is never an isolated motor act. It is rigidly bound into the near triad, which is coordinated largely by circuitry in the mesencephalic reticular formation.
The near triad. So when the vergence command is generated, it is automatically accompanied by two parasympathetic reflexes driven by the Edinger-Westphal nucleus.
Yes.
Walk us through those two reflexes.
First, you have accommodation. The ciliary muscle contracts, which releases tension on the suspensory ligaments of the lens.
And the lens passively thickens.
Right. It increases its refractive power to focus the diverging light rays from the near object onto the retina.
And the second reflex?
Pupillary constriction. The pupillary sphincter contracts, reducing the aperture of the eye.
Which increases the depth of field and sharpens the foveal image, effectively compensating for any slight errors in the accommodation reflex.
The near triad is a beautiful example of how the somatic motor system, so skeletal muscle controlling the globe and the autonomic nervous system, smooth muscle controlling the lens and pupil, they must fire in perfect synchrony to maintain a coherent sensory state.
It's brilliantly engineered. And this level of coordination brings us to our final challenge for the day: integrating gaze control with the movement of the entire head.
Right, which brings us back to the car.
Exactly. If you are driving and you need to check the side mirror, you don't just execute a massive 60-degree saccade, you physically turn your head.
But the biomechanics of the head and the eye are wildly divergent.
Because the eyeball has incredibly low inertia, whereas the head is massive and surrounded by heavy musculature.
So when you initiate a combined eye-head gaze shift, the motor command is sent to both systems simultaneously.
But because of the low inertia, the eye rapidly accelerates, executing a saccade that lands on the mirror long before the head has completed its rotation.
Right. And the problem arises the exact moment the head does start moving.
Because of the vestibular system.
Exactly. The semicircular canals instantly detect the angular acceleration of the head.
And this sensory input triggers the vestibulo-ocular reflex, the VOR.
Which is a brutally fast 3-neuron arc designed to stabilize vision by driving the eyes in the exact opposite direction of head rotation.
In a normal setting, if you are looking at a fixed point and shake your head, the VOR counter-rotates your eyes perfectly to keep the fovea locked on the target.
But during a gaze shift to the side mirror, the VOR becomes an active impediment.
Because your eye has already reached the mirror. As your head continues to turn toward the mirror, the VOR is violently triggered, attempting to counter-rotate your eyes away from the mirror back toward the center of the windshield.
Right. If the VOR were allowed to operate unchecked, your eyes would reach the target and then instantly be swept backward by your own head movement.
So the brain must actively, dynamically suppress the VOR during the actual gaze shift.
And this suppression is heavily mediated by the cerebellar flocculus again.
What exactly is the cerebellum doing there?
The cerebellum recognizes the corollary discharge of the intended head movement, and it computes a cancellation signal that perfectly neutralizes the vestibular drive at the level of the vestibular nuclei.
So it just turns the reflex off for a split second.
Exactly. And the moment the head movement is complete and gaze is stable on the mirror, the cerebellar cancellation is lifted, and the VOR is re-engaged to stabilize the new visual scene.
The sheer computational bandwidth required to execute a simple glance in a side mirror is staggering.
It really is.
I mean, in the span of a few hundred milliseconds, you are constructing parietal priority maps, selectively disinhibiting the basal ganglia, aligning sensory and motor vectors in the midbrain, integrating velocity into position in the pontine reticular formation, driving 12 specific muscle actuators across varying states of biomechanical tension, and actively canceling one of the most deeply ingrained vestibular reflexes in the central nervous system.
And this symphony of neural processing occurs entirely below the threshold of our conscious awareness. It executes perfectly, multiple times a second, allowing us to safely navigate a ton of metal at highway speeds.
It's humbling, really.
Yeah.
So we've traced the neurobiology of gaze, from the geometry of the orbital pulleys, through the catastrophic failures of the cranial nerves, down into the mathematical pulse step engine of the brainstem, and back up through the cortical architecture of pursuit, vergence, and spatial intention.
It is a closed system that relies on constant corollary discharge and massive sensory suppression, just to maintain the illusion of visual stability. We know the molecular kinetics of the neural integrator. We know the precise topography of the collicular maps.
Yet, as Candle's text implies at its boundaries, a profound mystery remains. We possess a near-complete map of the physical hardware and the algorithmic software that dictates how the fovea is redirected.
Right. We can trace the electrophysiological cascade from the visual cortex all the way down to the lateral rectus.
But the genesis of the primary intent, the mechanism by which the conscious mind freely selects a specific target from a chaotic environment, determining what holds true behavioral value, that remains completely elusive.
It is the intersection where the biophysics of motor control meets the philosophy of conscious will.
A perfect place to leave it. As you continuously sample the road ahead today, consider the unseen machinery dictating your reality. Keep studying the networks, and we'll see you in the next deep dive.